So we have one more sorting issue to deal with, and that concerns the mitochondrion, which we know has a whole bunch of proteins in here, the respiratory chain, ATP synthase. TCA enzymes are here. There's a lot of transporters that catalyze nutrient exchange and ion transport here. There's a bunch of proteins here, a bunch of proteins here. Now there's a small circular genome here with a handful of genes on it. And there is in fact a functioning secretory pathway that handles the insertion or secretion of proteins. And that reflects the fact that this used to be a bacterium. But the genome is so small, there's only a handful of genes on it. So the huge majority of proteins that are here, 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 or here are actually encoded by the nucleus of the eukaryotic cell and then wind up at their particular location. And that involves a sorting mechanism that's nothing like what we've talked about so far. So to give a quick overview of the solution to that problem from a eukaryotic standpoint, we can draw a eukaryotic cell around the mitochondrion and then put the nucleus here, and there's the DNA that's going to have the genes for all of those mitochondrial proteins. So it's transcribed. The RNA ends up in the cytoplasm where it encounters ribosomes like it always does. And those ribosomes translate a full-length pre-protein that's destined to the mitochondrion. That protein is full-length but immature. And I'm hoping when you hear those terms, you immediately think about what might keep something full-length and immature, and that would be an HSP70. And if you think that, you would be absolutely right. But this is a specific HSP70 whose job it is, yes, to maintain a full-length protein in an unfolded state stably for a short period of time. But its other job is unique to this particular type of HSP70, and that again points to the reason why there needs to be a family of HSP70s related but distinct in their jobs. In this case, this HSP70 is a escorter as far as the location of that protein, so it actually brings the unfolded protein to the surface of the mitochondrion so that the next steps can take place. And those next steps are shown on this diagram, figure 1222 from your text. So the HSP70 is not shown on this diagram. It will be shown on the one that's coming up after it, so it's going to come back again, but it's here at this point. What is added to this diagram is the existence of something that is not really a signal sequence, it's a mitochondrial targeting sequence, is the more appropriate thing to call it. And it is what is recognized by the HSP70, the cytoplasmic HSP70, to bring the unfolded protein to the mitochondrial surface in the first place. So it's also instrumental, that sorting sequence, in the first encounter on the outer membrane of the mitochondria. That is a receptor built into something that is called TOM, and this is translocon of the outer membrane, which is what this is, the outer membrane. This is the inner membrane, so you can bet that there's going to be TIM coming on. But in any case, this translocon is not related to any of the other translocons we've talked about, other than it does exactly the same function. In this case, it forms a pore across the outer mitochondrial membrane. So keep in mind the overall process has got to account for the insertion of proteins here, soluble proteins in the inner membrane space, integral membrane proteins in the inner membrane, and soluble proteins in the matrix. So it's got to be much more complicated than what we've encountered so far. So TOM does what it sounds like it would do. It serves as the translocon through which the unfolded, I guess we can call it a nascent chain, goes into the inner membrane space. This is one of several pathways for the sorting of mitochondrial proteins. In this case, the protein as an unfolded thing goes all the way through into the matrix. And if you're thinking, oh, what brings it there, I bet you can tell me what is going to be sitting right here. It's going to be in the next diagram. But in any case, it is driven into the matrix or dragged into the matrix where it looks like this. And then in the case of some of the soluble TCA enzymes, say, proteins, that sorting sequence gets cleaved off and degraded, and you're left with the mature protein. But now, as we mentioned, there's a bunch of proteins here, there's a bunch of proteins here, and a bunch of proteins here, and that mechanism is not provided for with this diagram. So what in fact goes on is that there is an additional sorting sequence built into many of the mitochondrial proteins in their cytoplasmic pre-form. So we could stick a little extra sequence, say, over here somewhere, which would be here on this diagram, and here on this diagram, and here at this point. And remember, I mentioned that there is a functioning secretory pathway in the matrix of the mitochondria left over from its free-living bacterial days. It can recognize this as what we would recognize as a standard signal sequence now, and insert that green thing into this membrane or secrete it into the intermembrane space. So that would be a means of accounting for proteins that are here and here. That is just a slight elaboration on this pathway. But there are also proteins here, and there are other ways of getting proteins here and here, and they tend to occur right at this stage, maybe even before the involvement of TIM. So it's possible for green things to come in here, and then in their secretory pathway as soluble things right here. It's possible for them to migrate out of TOM and become integral outer membrane proteins, and it's also possible for them to migrate out of TIM and become integral inner membrane proteins. And then this final diagram has a little bit more details covering essentially the same process. So what's going on here is exactly the same thing, only we have now those cytosolic HSP70s associated with the protein in its cytoplasmic form before it gets to the mitochondrion. And then there's the translocation through TOM, its attachment, the protein's attachment to TIM, and an additional ingredient to the process has to do with the energetics of bringing this thing through. Notice that there's no ribosome pushing stuff through, so everything has got to be a drag in this case. So because this is the mitochondrial inner membrane, it has the P side and the N side from our respiration days. The respiratory chain has pumped all those protons out, so they're right here, and this allows sort of the electrophoresis of this protein into the matrix of the mitochondrion using that pre-existing very large proton motive force. And then the other impetus for movement into the matrix is yet another HSP70, in this case it's the mitochondrial one, that is waiting right out on the matrix side of TIM, and it does the standard issue HSP70 reaction cycle, which, because of its location, is a Brownian ratchet. So that's going to ratchet the green soluble protein into the matrix, providing the energy, using the energy of ATP hydrolysis and the Brownian ratchet mechanism to pull this thing through.